A team of Italian geneticists opened a laboratory freezer in Padua in early 2026, pulled out a set of sample vials that had been sealed since 1978, and ran a sequence they had run once before, 11 years earlier. They expected static, old degraded fragments, background noise, nothing worth a press release. What came back instead made them rerun the entire analysis 3 separate times before anyone was willing to write it down. Woven into the deepest fibers of the Shroud of Turin, underneath the wound marks, underneath the centuries of candle smoke and pilgrim hands, was a genetic signature that didn't belong to any single human being at all. Some of it was human, ancient, scattered across bloodlines from 3 different continents. And some of it wasn't human in any sense a biology textbook would recognize. Colonies of salt-loving microorganisms that shouldn't be able to survive on dry linen, folded into the same threads that carry the image of a tortured man. Nobody in that lab was expecting to reopen one of the most examined religious relics on Earth. The Shroud of Turin had already survived a century of chemical testing, photographic analysis, and one deeply controversial radiocarbon date that was supposed to have closed the case for good back in 1988. Every few years, someone announces new equipment, a new method, a new claim to finally settle it, and every few years, the cloth somehow gets stranger instead of simpler. This time was supposed to be different. This time, the technology was sensitive enough to read genetic fragments that older sequencers couldn't even detect. And that's exactly what makes the 2026 result so hard to dismiss as noise. By the end of this investigation, you're going to understand exactly why scientists who have spent their entire careers being professionally skeptical of miracles keep walking away from this cloth with more questions than they started with, and why the DNA itself may be the most contested piece of evidence yet. To understand why that Padua result mattered, you have to go back further, to the last time anyone thought this mystery was actually going to get solved for good. October 1978, Turin Cathedral, northern Italy. A 14-foot strip of pale linen folded for centuries inside a silver reliquary, unrolled under laboratory lighting for the first sustained scientific examination in its documented history. Waiting around it were roughly 30 scientists, physicists, chemists, forensic pathologists, and imaging specialists, granted a 5-day, 120-hour window by the Archdiocese of Turin to do one job: figure out what the image actually was. Among them stood a photographer named Barry Schwartz, brought on as the team's official documenting photographer. Schwartz was Jewish, and by his own later account, deeply skeptical that he was about to spend a week documenting anything more than a clever medieval forgery. He expected pigment. He expected brushwork. What he spent 5 days searching for, under white light, under ultraviolet, under infrared, across every square inch of that cloth, he never found. No paint particles, no binding medium, no brushstrokes under magnification. The image sat only on the very top layer of the linen fibers, a coating measured in fractions of a micron, thinner than a single human hair sliced lengthwise. Scrape that surface with a blade and the color disappears entirely, leaving plain white thread underneath. In 4 decades of documented forgery techniques, nothing produces a mark that shallow. It's worth being honest here because not everyone on that team agreed with each other, and that disagreement still matters. A chemist named Walter Macrone, working independently from tape samples, concluded the image was consistent with iron oxide and vermilion pigment applied by a medieval artist, and he never backed down from that position. Most of the STIRP team disputed his methodology and his conclusions, and the disagreement between Macrone and the rest of the group turned into one of the longest-running feuds in the history of relic science. That argument, unresolved to this day, is part of why the Shroud has never been declared settled in either direction. Long before any of those scientists arrived in Turin, the image on that cloth had already produced one of its strangest moments in a darkroom in 1898. An amateur photographer named Secondo Pia had been granted rare permission to photograph the Shroud during a public exhibition, and when he developed his glass plate negatives that night, he nearly dropped them. The negative showed a normal positive photographic image, clear, lifelike, almost luminous, which meant the Shroud itself, the actual cloth hanging in the cathedral, was functioning as a photographic negative, a light and dark reversal that shouldn't exist on a piece of fabric centuries before photography was invented. Pia was accused of faking the plates, dragged through a public scandal, and largely vindicated only decades later when a second photographer, Giuseppe Enri, reproduced the same negative effect in 1931 under tighter controls. That single accidental discovery is the reason anyone thought to run the image through a NASA terrain analyzer in the first place, and it's part of why, by the time Barry Schwartz and the rest of the STIRP team walked into that cathedral in 1978, the cloth already had 80 years of unresolved optical mystery attached to it. There's also a documented history worth knowing before the science gets stranger, because it explains why so many separate research teams keep circling back to the same 14 feet of linen. The earliest confirmed historical appearance of this exact cloth is in Lire, France, around 1354, in the possession of a knight named Geoffroy de Charny, with no verified paper trail connecting it to anything earlier. It survived a documented fire in 1532 in Chambéry, France, when the silver reliquary holding it partially melted, scorching the folded linen and leaving a symmetrical pattern of triangular burn holes still visible today, patched afterward by poor Clare nuns with fabric squares sewn directly onto the damaged sections. It moved to Turin permanently in 1578. Every one of those documented events, the fire, the repairs, the centuries of public display, the handling by clergy, royalty, and pilgrims, is a plausible source of contamination, which is exactly the kind of contamination the DNA researchers would eventually have to untangle 400 years later. What genuinely unsettled the STIRP team wasn't just the absence of pigment. It was a machine called the VP8 image analyzer. 2 years before the Turin examination in 1976, physicists Eric Jumper and John Jackson were testing equipment at Sandia Laboratories, originally built to convert satellite imagery and X-rays into readable topographic data. On a whim, they fed a photograph of the shroud into the device. Normal photographs, when processed this way, come out as a warped, meaningless mess, because ordinary light and shadow in a photograph aren't real distance measurements, they're just brightness values. But the shroud image didn't distort. It resolved into a coherent, three-dimensional relief, a human form with a nose, cheekbones, and the contour of a beard rising correctly out of the linen, as though brightness on the cloth actually corresponded to the physical distance between the fabric and a body. That single result is the reason the Shroud of Turin research project came into existence at all. Jackson and Jumper were so unsettled by what they saw that they built an entire scientific team around a single test result from a device meant for reading satellite terrain. Critics have since argued that skilled medieval relief artists, working from a real corpse or a sculpted death mask, could plausibly encode comparable depth information by hand. And that argument has never fully gone away either. But no one, forger or scientist, has ever managed to reproduce the effect starting from scratch. Then there's the blood, and this is where the story stops being about optics and starts being about a body. For years, skeptics pointed to the blood as the weakest link in the authenticity argument because it looked wrong. The stains are a vivid, almost fresh-looking red, and basic forensic science says that shouldn't happen. Dried blood oxidizes and darkens within hours, and blood sitting on cloth for centuries should be black, brittle, and long gone. Bright red blood on a 2,000-year-old artifact looks, at first glance, exactly like what a forger with a jar of pigment would produce. A biochemist named Alan Adler, a specialist in porphyrins who spent decades studying the exact chemistry of blood breakdown, tested that assumption directly instead of accepting it. Working alongside physicist John Heller, Adler published a 1981 paper in the Canadian Society of Forensic Science journal identifying elevated concentrations of bilirubin bound into the shroud's blood residue. Bilirubin is a yellow bile pigment your liver dumps into the bloodstream in large quantities under extreme physiological stress, and specifically under the kind of catastrophic trauma associated with prolonged violent scourging. When bilirubin binds with degraded hemoglobin, the resulting compound resists the normal darkening process and can stay reddish for an extraordinarily long time. Adler and Heller's conclusion wasn't mystical. TE was clinical. The chemistry of the stains was consistent, in their words, with the blood of a man who had been severely flogged and crucified, not with paint, not with plant dye, and not with any pigment recipe known from antiquity. Other forensic work built directly on top of that finding. French surgeon Pierre Barbet, examining the wound pattern decades earlier, had already mapped over 100 individual scourge marks consistent with a Roman flagrum, a multi-tailed whip tipped with metal or bone designed to tear skin rather than simply bruise it. The wrist wounds don't align with the palms, the way centuries of religious art depicted them. They align with a small anatomical gap called the space of Destot, the only point on the wrist that could support a suspended body's full weight without tearing straight through, a detail no medieval artist is known to have understood or depicted correctly anywhere else in surviving religious art. There's a puncture wound in the side, consistent in width with a Roman infantry lance. None of this proves who the man was, but it describes, in unnerving anatomical detail, exactly how he died. And crucially, the shroud isn't the only piece of cloth making that claim. In the Cathedral of Oviedo, in northern Spain, a smaller blood-stained piece of linen called the Sudarium of Oviedo has a documented ownership history stretching back to at least the 7th century, with no connection to Turin and no plausible way the two objects were ever forged together as a matching set. Forensic researchers, who compared the two independently starting in the 1960s, found that the blood-stain patterns on the Sudarium align, point for point, with the wound positions described on the shroud, the same nose and beard bleeding pattern, the same blood type, group AB, first identified on the shroud by forensic pathologist Pier Luigi Baimaboloni in the early 1980s, and later corroborated on both cloths by other independent laboratories. Two separate textiles with two separate ownership histories, converging on the same wound geometry and the same rare blood type is not, on its own, proof of anything supernatural. But it is exactly the kind of independent corroboration that a lone forger working in isolation would have had almost no way to fake across two different cities, centuries apart. It's also worth mentioning what didn't hold up, because an honest account of this case has to include its dead ends. In the 1970s, a Swiss criminologist named Max Frey collected microscopic pollen samples from the shroud's surface using adhesive tape, and claimed to identify grains from plants native only to the region around Jerusalem. Evidence, he argued, that the cloth had spent real time in first-century Judea, years that pollen data was treated as some of the strongest circumstantial support for authenticity. Then, in the early 1980s, Frey was revealed to have authenticated the forged Hitler diaries as genuine historical documents, a catastrophic professional embarrassment that seriously damaged his credibility as an independent authority and cast a long shadow over his earlier, unreplicated pollen work. Later researchers were never able to fully verify his original tape samples or his identification methods, and most serious shroud researchers today treat the pollen evidence as unreliable at best. It's a useful reminder that this entire field is full of claims that sounded airtight for a decade and then quietly fell apart under scrutiny, which is exactly why the newer DNA evidence has been treated with so much more caution before anyone was willing to publish it. Which brings us to the genetics and to why a plain forensic mystery turned into a full-blown scientific controversy. In 2015, a research team led by geneticist Gianni Barcaccia at the University of Padua published a study in the journal Scientific Reports examining dust and fiber particles vacuumed from the shroud back in 1978 and 1988. They weren't hunting for a single donor's DNA. They were mapping everything present, plant material and human genetic material both, because both tell a story about where an object has physically been. The plant DNA alone was strange enough. European spruce, Mediterranean clover and ryegrass, North American black locust, a tree that didn't exist outside the Americas until European colonization, rare East Asian pear and plum species, a single 14-foot piece of cloth carrying botanical fingerprints from 4 different continents, which by itself tells you this object has been unrolled, displayed, and handled across an enormous geographic range over a very long time. Then came the human mitochondrial DNA, the genetic material passed down almost unchanged from mother to child, and this is where it gets genuinely uncomfortable for a clean authenticity story. Barcaccia's team identified multiple distinct haplogroups scattered across the sampled dust. Some markers matched populations from North Africa. Some matched lineages found in East Africa. Some matched genetic signatures still traceable to communities in China. And repeatedly, the oldest, most degraded fragments, the kind of DNA that has had the most time to break down, kept tracing back to lineages still concentrated today around the Middle East, the Caucasus, and northern India. Barcaccia told reporters directly that one of the most common haplotypes recovered from the cloth is still, today, disproportionately common within the Druze community, an ethno-religious population concentrated in modern Syria, Lebanon, Israel, and Jordan. He went further, noting that the shortest, oldest genetic fragments recovered pointed toward lineages typical of India, a detail that lines up unexpectedly well with the historical Latin and Greek word for the fabric itself, sindun, believed by some linguists to derive from Scindia, an ancient term for fine cloth associated with the Indus region. None of this identifies a single body. What it does, according to Barcaccia, is describe a well-traveled object, a cloth that has physically passed through or been touched by people across an enormous swath of the ancient and medieval world. Exactly the kind of contamination pattern you'd expect from a relic paraded through churches, monasteries, and private chapels for hundreds of years, not from a controlled museum specimen kept in a sealed case. Barcaccia was careful, in interviews at the time, not to overclaim what any of this proved. He described the diversity of species and lineages recovered as genuinely unusual compared to similar forensic textile studies, which typically show contamination from a much narrower, more local set of sources. A cloth that had spent its entire documented existence inside a handful of European churches should show a tight regional genetic and botanical footprint. This one didn't. And that mismatch between the object's known paper trail and its actual biological trail is precisely the kind of anomaly that keeps serious researchers from simply filing the whole case under settled forgery and moving on. For a long time, that 2015 study was where the genetic story ended, filed away as an interesting but inconclusive footnote next to the far more famous radiocarbon dating controversy. In 1988, 3 independent laboratories, Oxford, the University of Arizona, and the Swiss Federal Institute of Technology in Zurich, all radiocarbon dated a small corner sample of the cloth to somewhere between 1260 and 1390 AD, comfortably inside the medieval period and comfortably outside the lifetime of anyone living in 1st century Judea. The 1988 test was conducted under strict protocol, 3 labs working blind on coded samples supplied by the British Museum, and at the time it was presented to the public as essentially unimpeachable, a rare case of total scientific consensus across 3 independent institutions in 3 different countries. For over a decade, that result was treated by much of the scientific establishment as the final word. Then, in 2005, chemist Raymond Rogers, a member of the original 1978 STIRP team, who had spent 30 years defending the radiocarbon result, published his own reanalysis and reversed course. Rogers found that the specific corner sampled for the 1988 test showed clear chemical evidence of having been rewoven, patched with cotton fibers, and dyed to visually match the surrounding linen, consistent with documented repairs made to the cloth after a 1532 fire in Chambéry, France, that scorched one edge of the fabric. He also measured vanillin loss in the flax fibers, a chemical marker that decays at a roughly predictable rate, and calculated an age far older than the radiocarbon sample suggested, though nowhere near precise enough to function as a standalone dating method on its own. Rogers' findings didn't overturn the 1988 result outright, and the debate over whether that corner sample was contaminated remains genuinely unresolved among specialists. But it meant the single cleanest piece of evidence against authenticity had a real documented peer-reviewed crack in it. And that crack is exactly why researchers kept coming back to the cloth with better tools instead of treating the case as closed. Which returns us to that Padua freezer in early 2026 and the follow-up study that made 3 geneticists rerun their own results. Using the same official 1978 and 1988 sample collection, but now applying modern high-sensitivity metagenomic sequencing technology that didn't exist a decade earlier. Barcaccia's team, working again with longtime collaborators Alessandro Achilli and Antonio Tironi, went back into the dust. What they found first was almost anticlimactic. Multiple mitochondrial haplogroups again, including H2A2, the lineage tied to the standard human reference genome, H1b, common across Western Eurasia, and the rarer H33, still found today in pockets of the Near East. But then came a detail that reframed everything. One of the strongest, cleanest genetic signals recovered from the samples was haplogroup K1A1B1A, a subclade strongly associated with Ashkenazi Jewish ancestry. And when the team ran a kinship comparison, that exact haplotype turned out to match closely the mitogenome of the late Professor Pier Luigi Baima Belloni, the forensic pathologist who personally collected many of the 1978 and 1988 samples and who had Jewish heritage himself. In other words, a meaningful portion of what looked on the surface like ancient genetic evidence from the man depicted on the cloth was, at least in part, the researcher's own biological residue, left behind decades ago during collection and handling, and only now precise enough to identify by name. It's a strange kind of anticlimax, and also a genuinely important scientific correction, a reminder of exactly how easily modern contamination can masquerade as ancient signal, and exactly why every claim about this cloth has to survive being checked twice. But the kinship match wasn't the part that actually startled the lab. Running alongside the human genetic analysis, the 2026 team reconstructed what's called the metagenome of the cloth, essentially a full inventory of every microorganism whose genetic trace could be recovered from the fibers. Most of it made immediate sense. Bacteria and fungi commonly found on human skin, the ordinary biological residue you'd expect from centuries of hands, breath, and candle smoke near a heavily venerated object. But mixed into that expected human microbiome was something that didn't fit the environment at all. Genetic signatures from halophilic archaea, an ancient domain of single-celled organisms that require extremely high salt concentrations to survive. The kind of environment you find in salt flats, brine pools, and heavily salted preserved materials, not on dry linen stored inside cathedral vaults for 2,000 years. Alongside them, traces of fungal genera, including species related to Debaryomyces hansenii, a salt-tolerant yeast more commonly associated with cured meats and briny food preservation than religious textiles. Nobody in that Padua lab is claiming these organisms prove anything supernatural. The leading working explanation is mundane on its surface. Ancient burial practices in the region sometimes involved packing bodies, or wrappings with salt or salt-rich substances as a preservative, and if genuine organic material from a real burial did come into contact with this cloth at some point in its documented or undocumented history, a resistant, dormant population of salt-loving microbes could plausibly have hitched a ride and survived, essentially fossilized in place, for far longer than anyone would expect. It's the kind of detail that sounds small until you sit with it. Whatever this cloth actually is, something about its earliest chemical history matches the exact preservation conditions you would expect from an ancient burial, not from a 14th century artist's workshop. So where does that leave the headline? The idea that some of the DNA recovered from the Shroud of Turin isn't human at all. It's true, but not in the way a viral thumbnail wants you to believe. Nobody found alien genetic code. Nobody found an unclassifiable species. What researchers actually found, layered into the same fibers that carry a three-dimensional wound pattern no forger has ever successfully replicated, is a genuinely strange mixture. Human mitochondrial lineages scattered across 3 continents and 2,000 years of handling, contamination from the very scientists who tried to study it, and a population of salt-dependent microorganisms that has no obvious business surviving on a piece of medieval or ancient linen at all. Every one of those findings, taken individually, has a plausible, non-miraculous explanation. Taken together, stacked on top of a wound pattern encoded at a resolution no known forgery technique from any century has ever matched, a blood-stained chemistry that correctly predicts trauma symptoms nobody in the medieval period understood well enough to fake, and a radiocarbon date with a documented unresolved contamination problem, the picture stops looking simple. Barry Schwartz, the photographer who flew to Turin in 1978, expecting to spend a week exposing a fraud, spent the rest of his life instead cataloging every scrap of Sturp's data for free, publicly, so anyone could check the work themselves. He said, on record, that he went looking for evidence of a hoax and came home with something he still can't fully explain. Nearly 50 years and 3 separate genetic studies later, neither can the scientists who keep going back for another look. Every generation of new equipment was supposed to be the one that finally closed this file. The VP8 in the 70s, the radiocarbon labs in the 80s, the pollen analysis that followed, the first genetic study in 2015, and now a metagenomic sequencer, sensitive enough to read individual microbial genomes out of 40-year-old dust. Each one added detail. None of them delivered the clean, simple verdict anybody actually wanted, forgery or authentic closed either way. Instead, every new tool just adds another layer of evidence that has to somehow fit inside the same, increasingly crowded, increasingly strange picture. Here's the question worth sitting with. If the blood chemistry independently confirms catastrophic torture consistent with 1st century Roman execution methods, if the image sits on a layer of linen too thin for any known pigment to produce, and if the genetic record shows repeated, unexplained contact with an ancient burial environment that modern archaeology can trace, but modern chemistry still can't fully replicate, what exactly are we looking at? A masterwork of medieval forgery so advanced it accidentally anticipated forensic science techniques that wouldn't exist for another 600 years? Or a genuine, badly degraded, heavily contaminated piece of physical evidence from an execution that happened 2,000 years ago, dragged through centuries of well-meaning human hands that kept unintentionally scrambling the very data that might someday prove what it is? Type case closed if you think conventional forgery still explains everything here. Type still unexplained if you think the evidence has moved past that. And if this is the first time you've seen what the actual peer-reviewed research says, instead of the version that gets flattened into a headline, stick around, because we go back into cases exactly like this one every single week. Cold, forensic, and grounded in the paper trail instead of the legend.